Okay, good afternoon. Welcome. It's my distinct privilege to welcome all of you to the Reitz Visiting Professorship Lecture. This is being held in conjunction with the Stanford Cardiovascular Institute, and therefore on behalf of my close colleague and partner, Joe Wu, WU, it's really our honor to be introducing Dr. Bart Griffith. So first, I just want to say a word. You hear me? Yeah, better? No? I think today we have two individuals in the audience that define the word pioneer. Dr. Bruce Reitz started the entire field of heart-lung transplantation right here at Stanford. Many of you may not recognize that this brand new operation was a procedure that Dr. Reitz developed in the research lab. Just like many of you are studying new novel pathways, technologies, Dr. Reitz went into the lab with a dream, a novel idea, and studied this and perfected it, and then eventually translated this into the care of a human being, and very successfully. That very first patient of his lived 5 years after that landmark transplant. And that operation has been done throughout the world and saved so many lives. And so I think that is the definition of pioneer. And it's so appropriate that under Dr. Reitz's name, we're able to welcome Dr. Bart Griffith, who, as many of you know from reading CNN and various newspapers, did the world's first xenotransplant successfully into a human being of a genetically modified pig. And we wanted to have Dr. Griffith present this. And since there's so much science here, we thought we'd have to do this not just in front of the Department of Cardiothoracic Surgery, but we'd have to bring the real scientists of the CVI out to question him. So no holds barred on the inquisition afterwards. He's ready for the challenge. Dr. Griffith is a world-famous surgeon who is intimately involved in artificial heart devices, transplantation of all sorts, and most recently, xenotransplantation. And we're here today to learn a bit more about that extraordinary journey from the lab into animals, and then into human beings, and maybe learn a little bit about the tremendous impact yet to come for generations of people down the road. Dr. Griffith, thank you so much for coming.
Thank you. Thank you for being here. How's lunch? Pretty good, I hope. It's great to see Bruce. I haven't seen Bruce for a few years, and it's really, really nice. And I'm going to say a few words about Dr. Reitz as I get warmed up here to talk to you about the most recent project of pig to human heart. It's great to be a cardiac surgeon, actually. It's still great to be a cardiac surgeon when I can't hear. It's on. Hello? Hello? Want me to use the microphone? Okay. Hello? Is it better? All right. Sorry. It's great to be a cardiac surgeon. It was great when I was in training, when I was young in faculty, and now that I'm old in faculty, very old in faculty, it's still great. I met with the cardiothoracic surgical trainees, some of which came to lunch yesterday, the others of which I have their cards, so I know who didn't show. But it is wonderful to see their brightness and their passion for healing. And it's all about that. It's hard work, but things just kept evolving. And this is a little bit of arc of career in terms of what I was able to experience. And when I was a trainee sitting in the audience, none of this existed. it's amazing what's happened. And just touring your campus today, some of the deep science, that is going to be impactful. It's just hard to deny how exciting it's going to be. And right away, the wheels start to crank out how I can appropriate this to surgical science. And I wish you all the very best. You're here because you're bright, and I hope you're here because you have empathy, which made all the difference for me. The bottom two organoids, if you wish, are there because I do think that while we've made great progress with standard transplantation, we will see, organs on a chip. On the left, that's a soft lung that's been created, and on the right side is just a lung that's been decellularized, awaiting to be recellularized with human cells. And it's really literally going to be within the scope of your early practice as a student to see that. Xeno is a buzz right now. The world is picking it up a little bit. These are Chinese investigators that made news early last week by placing a liver as a bridge to transplant in a decedent, but in a bridge to transplant kind of method in China. So they have their own genetically altered organ, and they are a tour de force. Our colleagues at the MGH made big news, putting a kidney in a live patient. So that would be the third patient that had a gene altered organ for the purposes of transplantation. It's pretty incredible. It's pretty terrific. And it's a great time to be alive. Bruce, I don't know whether you remember this picture that you took, I think, and then sent to me. But this was a picture when I was lucky to be on a trip with Bruce and Dr. Bando. And we were, I think, on our way up to be at sunrise to the top of Mount Fuji in Japan. You stop at this little place, and I think you slept there so that you could be ready at 4 a.m. to climb the rest of the way. But this was the hut, and you took your shoes off. And of course, Bruce's shoes, I can show you right here. You tell me if I'm right, Bruce. I think those are your shoes, because I know those are my boots. Anyhow, there you go, Bruce. I think of you, and I think of the many steps that were taken to get to the top of Fuji, but it is obviously just a reference to the many, many steps it takes to get where you want to go, no matter what you're doing. And it's reminiscent of that. And there's Bruce and my son, Cullen, who's an orthopedic surgeon now, Bruce. And I don't see Jay in this picture, but he was with us. And then I'm on Bruce's left. So what a nice time that was. And it's really great, Bruce, to be here because you were the most welcoming in sharing your science and your encouragement. I followed Bruce's interest in heart-lung transplantation. About 5 months later, we both had cyclosporin, and he did all the work out of the operating room to get it into humans. And then he showed the way to the rest of us. And I hope you understand copy is the finest form of flattery, Bruce. And you made all the difference in a young surgeon's life and helped me take off. That's a patient with alpha-1 antitrypsin deficiency. I think Bruce created this for congenital heart affected patients because his passion was in the children who had Eisenmenger syndrome and couldn't really be corrected with standard surgery. So there we are. What's that? This is an old picture, and it came out of the playbook that the Stanford transplant program was using to monitor whether or not a rejection episode was going to happen. This is called an E-rosette, and they're red blood cells that are rosetting around those other cells, and those other cells just happen to be T lymphocytes. And in an era where we didn't have flow, where we couldn't really measure subsets of lymphocytes and didn't even know if we could what they did, this was very basic type of monitoring that came out of the Stanford program, and it was the standard of care up until 1980. And now I'm going to share with you some other things where we're now looking at bulk transcriptomics and really wonderful tools available to us to follow transplant patients to give better and more strategic immune suppression going forward. This is that lung being decellularized, and it's over time lapse here over a period of a couple of hours. And this is how it's done. That is basically an enzyme to remove all the cells in that lung. It becomes just a white matrix. And then the white matrix receives 3 cell lines, human cell lines, for the purposes of now creating a whole lung, which is allo, for transplantation. And of course, if we use cell lines from a particularly targeted recipient, perhaps this would not be allo anymore, it would be yours. It would be an auto lung transplant. So that's a lung from a swine, so it's a swine matrix with human cells. The big bugaboo in all this recellularization has been the vascular beds. And this is a picture of what currently is capable in the vascular recellularization. The tight junctions are beginning to become more often. And these lungs, when tested, look to be a little tighter, and they're not becoming edematous early, and we're getting good oxygenation. So I think it's going to happen. I think, again, Xeno sounds spectacular. Imagine growing a lung just for you. Okay, now we've got 4 hearts up there, if my count's right. And I might ask Bruce, because he's always a willing participant. This will be an audience participation. Bruce, there are 2 hearts up there from pigs, right? And there are actually 2 hearts up there in pigs that went into humans, and 2 hearts up there from pigs that went into baboons. And can you guess which one went into a baboon? Top one, and there's one more. Left one. Bruce was always at the top of the class. That's why I asked him. That is true. So it's obvious that the heart on the top is different than the other 3. The other 3 are thicker. And the ones on the bottom to the left are very thick. The top one came out of a 9-month recipient, baboon, from a Xeno pig heart. The heart had 10 genetic edits, just like the ones that we put into humans. Looks pretty good. That animal was taken from us from a viral infection that took down half of the whole colony. That heart had no evidence of rejection that we could determine at autopsy. So it looks pretty good. That's what it should look like if you take a heart out and you slice it across in a short axis view. The bottom left heart, rejected in the baboon. Now, baboons, when they go downstairs into their living quarters, if they get sick, they die pretty quickly, because we don't really have a way to resuscitate a baboon. They're wild animals. They don't tell you how they're feeling. They just start to not eat very well. They get slow, and then they pass. And they oftentimes will pass in the night. So it's not a long and strung out kind of thing. But this is an example of a baboon heart that's undergone antibody-mediated rejection. It's become a little swollen, and I'll explain why that happens. The bottom 2 on the right side are 2 human recipients, 100 days total, sustaining health, if you wish, in human patients. And those are autopsy examples of the hearts that had been removed after we'd placed them in. So those are the hearts that our 2 patients had. And they're very thick. And they got thick because they underwent antibody-mediated rejection. That's a capillary-based problem where antibodies blow up the capillaries, if you wish. And when you blow up your capillary, it's very similar to what happens when you walk off the curb in the winter, and you twist your ankle, and you notice all of a sudden, by the time you look at it, it's the size of an orange. And then it's the size of a grapefruit the second time you take a look at it. So swelling happens fast in injury, particularly when it's vascularly based. And that's exactly what happened to these hearts. They went into diastolic heart failure within 24 hours. They got swollen, they couldn't relax, they kept squeezing. We had pretty good systolic function until the very end, but they couldn't relax in diastole. And just like a bellows, if you can't relax a bellows, you can't get any air out of it when you squeeze it, right? So that was the physiology that we saw. And this is our call, Houston, we've got a problem. So the rest of this talk, I hope, is going to focus a little bit about patients and stuff, but it's going to get to the answer of what we need to do to fix this problem that was a little bit unanticipated from our non-human primate studies before we went into man. But it did happen. You notice there's one animal here that's very similar. And because the animal died sooner, we kept the humans alive longer, it just progressed more in the humans. That's why it's thicker. But you can see it's growing in that way relative to the top one. And the type of growth was remarkable. In 10 days, these animals doubled their heart rate. It was water. Okay, the pig heart's pretty special, and it gives us the tool that we can work with. And as you've heard, probably it's a genetically altered heart, and it works. This is our second recipient on the left, and on the right is his first postoperative radiograph. I don't think it takes a keen eye or a fourth-year medical student to tell us that the lungs on the right side are a little bit better than the lungs on the left. The heart is pumping better, and the edema in the lung is being removed early postoperatively. So you can see that's the shape of a pig heart in a patient. Looks pretty humanoid. Kind of a nice photograph. All of this started probably in, well, we can take it back to 2014 when Tom Starzl talked Martine Rothblatt into investing in this concept of creating organs from animals. And the work had gone on for maybe 20 years before that and began with Dolly. So the company that basically did Dolly, which was a black-faced sheep from a white-faced mother, you know, that was the first large mammal that had been cloned. And so with that, Tom Starzl, who at the time was still my mentor at the University of Pittsburgh, he saw the application as being possible for Xeno organs for transplantation. And so that collaboration has led to the current clinical 10-gene altered pig. UTHR is United Therapeutics, that's their trademark or whatever. And that's as crazy and nutty as the people running the show. They're really visionary, but they are sweet people to be around. They are lots of fun. If you take a wild heart, meaning not genetically altered, from a pig, and you sew it into the abdomen of a baboon just to see, you know, whether or not the thing is going to work for a while, as opposed to a working heart, it's just sewn into the vascular supply. Something happens remarkable very quickly. It turns black. Why is it turning black? Well, it's kind of like our reaction, like the bee sting. You have antibodies that are ready to go, and they basically lay down blood clot and complement and form thromboses in the microcapillary system. And so you have instant infarction. This heart was not protected in any way going into the non-human primate. It was just a wild heart. So the difference, of course, would be if you could knock out what it was that was priming those antibodies to cause that vascular reaction. Much of the important genetic re-engineering or alteration in the pig heart, in the pigs, relates to coagulation. And it relates primarily in the regulation of complement and the regulation of thrombomodulin and EPCR, which really regulates the thrombomodulin effect. Complement regulation is really, really important. And in a pig is insufficient. There's not enough complement regulator in the pig heart relative to human. And so it needs to be strengthened. The pig has CD46 and HDAF or CD55, just not enough of it. So it's added to it. And then the thrombomodulin is similarly not robust in the pig and needs to be strengthened. The two genes on the most right-hand side here deal with inflammation. Xenoreaction is an inflammatory process, and it was felt that if you could help tamp down regulatory factors using an antioxidase gene, HO1, and CD47, which is a migratory macrophage inhibiting factor. Those things would be very, very helpful. Growth control, pigs grow to 400 pounds. It was felt that organs shouldn't be growing to 400-pound type organs. And so they put a knockout on a growth receptor to create dwarf pigs, which is what we're using. And then finally, the most important knockouts, right? So we had these 6 knock-ins. Now we've got 4 knockouts. One was a growth receptor, and the other 3 were sugar sugar-based, carbohydrate-based epitopes that were recognized immediately by your antibodies and created that black heart that occurred in minutes that you saw in that wild heart. So if you could knock out all those gals and not have to deal with it, you'd be off and running. This is a immune histochemistry panel of those genes to show you that they're working, to show us that the heart that we put in did, in terms of the genetic transgene expression, that it actually was happening. So if you look at the panel, it's good to kind of probably combine it in two sets. So the bottom set are those genes that are expressed constitutively. They have a CAG promoter, and it's on all the time. As you can see through thick and thin, there's a heart biopsy on the left where the biopsy showed no rejection. And at the autopsy, the relative degree of staining was about the same. I assure you, it might not look quite the same to you because of differences in the areas we picked, but it was felt to be persisting and adequate transgene expression. This is a negative control in the middle column. If you look at the top two, they're very interesting to me because you can see thrombomodulin and EPCR. Again, those are things that hold back thrombosis. You might not want that transgene on all the time, you'd bleed, right? So that it's actually on an endothelial promoter, an endothelial specific promoter, and under stress, the endothelium will express it. And you can see that the first biopsy, when our heart was not under stress, this particular patient, it's not being expressed, but at autopsy, it's going bonkers, right? It's saying, I am in big trouble, my endothelium's in trouble, and I'm turning this gene on. So it's kind of a smart gene. It's really quite fascinating in terms of that. You can see that there's another way to express this, just to show the columns. You can see that the important thrombomodulin and EPCR were expressed only when the heart was in trouble. Okay, you with me? Good. So how do we get to humans? Well, we start like Bruce did with non-human primates. And this was what really linked it up for us. I showed you that wild heart going into the abdomen of a baboon. Well, the series that was put together by my partner, Dr. Mohideen, at the NIH, used the progressively engineered animal hearts in a situation where they wouldn't have to work, they just have to beat. And a beating heart was a heart that was a good heart, right, over time. So they were sewn into the abdomen, aorta and vena cava like this, PA into the IVC and aorta to aorta. That revascularized that heart. But again, that heart wasn't working, so it wasn't a true heart transplant. But you can see in the abdomen of this animal, a thing that's beating back and forth is non-aneurysmal. Don't go and get your endographs out. That's actually the heart beating. And it says day 784. This animal was one of the long ones. The longest was 3 years. So with that being accomplished, I think it was clear that the test next to be done would be to move it out of the abdomen, put it into the chest, and actually do the allo transplant or the xenotransplant in the baboon, right? You with me? So non-working to working before we could go to the FDA and say, please, can we go forward? And that's exactly what happened. We made progress with respect to the, over a period of 5 years, when Dr. Mohideen joined us at Maryland, he and I were able to be progressively more successful in our techniques and finally became 100% successful. We did lose an animal for like 25 implants in terms of operative loss. So worked out all that stuff and got to the point where we said to each other, we're ready for a multi-center trial, went to the FDA. They said, okay, give us 6 more done the same way, and you need to have 6 months to a year survival rate. We said, fine, that's all right, but what are we going to do while we're doing that? Because we're not allowed to change anything in the protocol. So instead of just watching ourselves do the same thing for 2 years, in essence, running on ice, we sat down and we talked about whether we'd be ready for a human. We had 9 months survival in the baboons already with the protocol that we were testing. And we thought if we had a human with no other option, we might go ahead and offer this particular procedure under the auspices of, in essence, a compassionate release, or what they call an EIND now, which is expanded EIND. The FDA views this heart as a drug. It falls under the biologics in the new CBER area, and therefore it unders the IND, which is new drug classification. So our patients have accrued a total of 100 days of survival, the two of them. This is a Raz Chast cartoon, which is kind of says it. This guy is saying the end is near, have an ice cream cone. I kind of doctored it a little bit, and I put myself and Dr. Mohideen there, and the end is near. You may as well have a pig heart. So it's kind of like what our patients ended up saying. And I don't mean this to be crude or unnecessarily taking advantage of the patients. They loved this. They all went through the period of time where they had to accept their deaths because they were dying and couldn't have a standard procedure. They would not leave the hospital without a heroic kind of experimental transplant. By the time we got around to explaining that the animal transplant was actually going to be a pig heart, they were saying, well, will I oink? They were humorous in terms of their response to it. They weren't told that they were necessarily going to survive, and that it was something that our ethicists felt very strongly about. We had an ethicist team, at the bedside all the time. We had 4 psychiatrists see our first patients because our dean couldn't imagine that anybody would actually agree to this. All the psychiatrists felt, in spite of the patient's illness, that he had a good grasp of what he was about to undergo. And our ethicist is shown there the morning after the first procedure with the black coat on. It's kind of weird and looks almost like, Dr. Death there, but he was with us the whole way. And I show this for lots of reasons. In some ways, gone are the days when we had this multidisciplinary rounding with excitement in the ICUs. We get the work done on the weekends. This is a Saturday morning. We had the whole team there, of course, one day post first Xeno transplant. But it was thrilling. I will tell you, it was really exciting. And it kind of got all the juices going in everybody. So everybody liked it a lot. The night we got approval to do that procedure is shown here. It was New Year's Eve at 5:30. I got this email from the FDA, and it's basically saying, Dr. Griffith, we've evaluated we were back and forth at 4 different iterations, and we all felt we weren't going to be approved. They approved us. The patient was getting really sick by that time, and that had tragic consequences, but nonetheless, was pretty interesting. Then, the night before the procedure, they kept asking us when we were going to go, and our hospital required another almost 12 days to get to yes, for a lot of reasons, so I won't go into, but unless you ask me. We wish you the best of luck tomorrow. That was pretty neat to get that email from the FDA. I mean, it was almost empowering to me, and it kind of took me off the hook because I figured there were 20 FDA investigators that put their soul into this review, and they said, go ahead. You've done the work. Go ahead. And it gave me license, in essence, to do what I thought was right, but many others might not have agreed to. This was a favorite. I got a lot. My phone obviously went cuckoo bird, and this was an email I got from Craig. I don't know whether Craig made it today, but he told me he might make it. But he sent this to me as Craig Miller, and it says, "Well done, compadre." That's definitely all Craig. And it says, "Bart, Dr. Shumway would be very proud of you," parentheses, "despite his general pessimistic outlook on the field, for doing xenograft heart the proper way after tons of years of experience, blah, blah, fingers crossed, best." Pretty neat. Those are the high points. You get those wonderful kind of suggestions, boom. So this was our patient, our first patient. I've shown this slide a number of places, but I think it's important to know how sick these recipients were. This gentleman was hospitalized for 61 days straight on his back, 48 of them on VA ECMO to support his heart and his end organs. His white count was always low. It would go as low as 1 with antibiotic treatment. His platelet count was also low. Very, very striking sarcopenia, best evidenced by the fact that my not so big size 7 glove hand can go all the way around the widest point of his lower extremity there. That's his left ventricle, and it'll beat for you here a little bit. It was huge, and certainly nothing that would be likely treatable with medical therapy. His sarcopenia is also evidenced postoperatively in his lack of chest wall musculature. I learned this whole word, sarcopenia. I love the word, but he had it. That pig heart was a rock star. Silence any criticism that the pig's not the best organ, you know, to put in a human. It's adequate. It may not be the best, but you'd have a hard time finding a better one. This is a stress echo, right? It's longitudinal stress we're looking at here, strain, and it just is off the charts in terms of its ability. I'm on some timer here. I don't know. Excuse me. So it did very, very well, and this was for up to 43 days. So we thought we were off and running. We were making plans to get the patient out of the hospital. There was a funny blip here, and this is a nod to the benefit of using new techniques for surveillance. And we were surveilling for cell-free DNA snippets of microbacteria and viruses. So we were using his blood, you know, instead of cultures, we were looking at the cell-free snippets, and we could pick up all kinds of different viruses, one of which was the pig CMV virus. And our ID guy comes up about, well, it was about 25 days. He says, you know, we've got a strange little blip going on here, and it's PCMV. And we all laughed. We said, well, we ruled that out with 4 PCRs. He doesn't have it. That's a latent virus. Comes out in the blood in the baby pig for maybe a week or 10 days and goes away. So it's hard to pick up with PCR. It's sitting in the cell, hanging out like a herpes virus, you know, waiting for your immunity to drop and come out. Well, this man was pretty immune suppressed, and it was ultimately shown to be accurate that this actually was PCMV, and it was in his heart, and it was growing. You could see that the amount that was being shed into the blood was increasing, and he gave some signs of that. And we all thought that this was a big problem. The problem was his antiviral therapy had to be abbreviated because of his low white blood cell count. And he was off MMF even for most of this period. So he went through a series of different treatments. Finally, we just said we have to give him the therapy. And when we gave him the therapy, you could see we knocked down the virus. But I think that the horse was out of the barn in terms of if it was going to damage the heart, it would have done that by now. The left side frame shows a pretty good looking short axis echo. That's when things were hunky-dory. And when he was a rock star heart, 49, he began to thicken. That echo on the right is a thicker heart. Not as thick as it will come to be, as you've seen on the autopsy, but it's beginning to change. We weren't sure what was going on. We did a number of biopsies, and going back serially in biopsies, that's the endomyocardial biopsy at day 30 is shown on the upper left. We didn't do it sooner because he had no platelets. So we were constrained in all ways by his bone marrow. But that's a normal endothelial cell. That's a real big nucleus of an endothelial cell, and I'm told that's normal. His second biopsy shows some swelling of the endothelial cell. You don't quite see the same luxurious nuclei. And then finally, the third biopsy shows dissolution of capillaries. Where do you think PCMV lives? Lives in the endothelial cell. So in essence, it's your lip relative to a herpes virus. And so if it were to replicate, it would come out of the capillary cell. So we had a number of people feeling that that's what happened to this heart, blew up his capillaries, the heart began to leak and became edematous. But when we really looked hard in the tissue, we saw very little evidence of the virus in the heart. There were some transcripts of replication in the right ventricle that we could find. We found no virus in any of the other organs, but we did find shed DNA of that virus. So the virus blew up with the heart when the heart blew up. And whether or not that was the problem, we were uncertain. We spent a long time trying to understand what had happened to the patient. We wrote the patient up in what we thought good articles in good scientific journals. They held us, our toes to the fire, so to speak. The second one on the right is in Lancet, and that basically was after a year of studying the tissues. The conclusion of the one on the right was not very different than the one on the left. After a year and a half, we still didn't know whether he'd had an antibody-mediated rejection or the virus caused the whole problem. So what did we do? We decided a second patient was in order after 2 years, and we began to try to apply the lessons. Better bone marrow in your next patient so you don't have to bridge your therapies. Make sure that you can rule out the virus that is difficult to rule out by nasal swab. We did that by using blood testing of a new assay, and we felt confident. Try to get a patient a little less sick. So this is our second patient. He came into the hospital. We had hoped to keep him at home prior to transplant, but he was admitted and had a cardiac arrest almost the day he came in. This was his admission chest x-ray. That was the one I had shown you already today. That was his CT scan. So he wasn't in great shape, right? We had hoped to avoid that. But there he is on his second postoperative day, looking like really almost any other patient in the hospital, standing, going to be up and about. This is kind of interesting. I hope we can get this, guys. How do I play that movie over here? Where do I? How do I make that movie thing there go? Should I do it here? Okay, I'll do it here. So, thank you, I'll take it. This gentleman was a histopathology technician at the NIH, so he understood medicine, and he would make slides. So he was used to looking at stuff. So after his first biopsy, we printed it out and brought it up to him.
Larry, what do you see in here? I see perfectly normal heart all the way across right there. Right, starting to break down. Yeah. So you're happy about it? I'm very happy about that. Great. That's exactly. Okay, so Larry, what do you see in here? Larry, what do you see in? Larry, what do you?
Sorry, everybody. Holy catfish. I'm so sorry. Everybody all right?
See in here. I see perfectly normal heart all the way across right there. Yeah. Right, starting to break down. Yeah. So you're happy about it? I'm very happy about that. Great. That's exactly what. Okay, so yeah. This one looks like completely normal heart. Yeah. That's definitely what we wanted to see.
Understand that man has a base heart. I mean, how could that be? So through his course, we were able to, again, look at the opportunities of having excluded zoonotic viruses, which were shown to be true. There wasn't anything that we were worried about in his follow-up, so that the preoperative screening was probably pretty good, and we had improved that piece of the program from the first patient. The PCMB is shown there as negative. PERV-A and PERV-V, we expect to see. We don't have PERV-C in the animals that's been bred out of them, so we don't have to worry about those extra 50 edits that were done in the Massachusetts pig. If you don't have PERV-C to recombine with A or B, they can't replicate. So that's another way around that problem. Anyhow, zoonosis seems to be not a big problem. So here he is. He's still hanging in there. He's playing some cards, doing pretty well. But then we got into a few problems with him over time, just like the first patient. And we began to see this worrisome trend where he seemed to be doing the same thing as the first patient and didn't have the virus. So one of the leading contenders of the first patient's problem, we no longer are thinking, is possibly the answer. Probably it was a rejection phenomena. And that would be antibody-mediated rejection. There's a series of top two lines. One is left ventricular ejection fraction in green, and his strain is in blue, and his left ventricular width, his posterior wall dimension is shown in the orange, and everything's going the wrong direction in time here. The heart's getting thicker, his EF stays pretty good for a while, and then finally bombs. And then his strain, how well he contracts on that echo, that rock star echo, became less impressive over time, once he got out past about his 30th day. We did some things to try to rescue him. We added eculizumab, which is another way to complement impede, and we used a B cell, really cancer drug, to try to reduce the amount of antibody-mediated process going on in him with carfilzomib. Those two drugs are likely going to be important in the future. This is what his first biopsy showed, even though he was telling us looks pretty good when we got the immune histochemistry 12 hours later or 8 hours later. We were aghast with the amount of IgG and IgM staining. We were shocked, even though the EM looked, or the H&E looked pretty good. He agreed. And we also saw heavy staining of complement. So those two hallmarks suggest antibody-mediated rejection. It was far earlier than we expected. He had had some bleeding, and he did receive fresh frozen plasma and a number of units of red blood cells that weren't washed, suggesting we could have actually transferred passive human anti-pig antibody into him that he wasn't generating. Because in fact, he had so much IgG, which is a late, there's actually a switching that goes on with IgM to IgG. He had a lot of IgG in that first biopsy. So we thought some of that might have been passive. His later biopsy down the road here begins to show progression of the rejection phenomena with red blood cells leaking into the interstitium, macrophages. But notice he doesn't have much T cell effect here. When Bruce and I were looking at a lot of these biopsies, it was always the T cell that was creating havoc. We had lots of inflammatory process. You'd hold the slide up and it looked like there was more blue than pink because of round cell infiltration. That is not what was happening here. H&E always looked fairly benign, relatively speaking, but it was all happening at the capillary level. You lose your capillaries, you lose your perfusion, you lose your heart. World has progressed from that rosette that I showed you early on. This is a picture returned to us within 48 hours of having sent it abroad to Alexander Lupi in Paris. He does bulk transcriptomic research on tissue. And this is a volcano plot, which basically shows everything on the right is a gene that's upgraded, and everything on the left is a downregulated gene. The genes, by and large, that were upregulated were those that we traditionally associate with antibody-mediated rejection. And this was this is a 13-day biopsy, I think. Yeah, this was at 13 days. So if we'd had it during the course of events, we learned this after he died. So it's part of our new armamentarium to use it contemporarily. But looking backwards, if we'd had it, we would have been far more nimble at treating that biopsy. We ended up not thinking that biopsy had much to really tell us in terms of treatment. When you talk about antibody-mediated rejection, it all happens with human anti-pig antibodies. And this patient's, the two patients we did here are shown here. One had a little elevation of IgG and the other had a little elevation of IgM to pig. We all have anti-pig antibodies because it gets into viruses and bacteria and we inhale them and we ingest them and our body reacts to them. So we all have anti-pig antibodies more or less. And just like some of us have HLA antibodies, these antibodies can also be against proteins that we don't delete. We delete sugars, which are like our blood groups. Pig also has HLA-like protein antigens, which are called SLA, and we're just getting into how to specifically match for those. And I think that's something that's going to happen for us to be more reliably successful. None of these patients pre-op had CDC cross matches to their pigs, which means in the mid-1970s, this was standard of the art in human allotransplantation to have a negative CDC crossmatch. It didn't tell us the story, though, did it? Because we clearly had problems that these tests did not predict. Antibody-dependent cytotoxicity is shown here. This is what happens, and this was that biopsy with all that IgG and IgM. The biopsy looks pretty good, but it's beginning to aggravate the endothelial cell in a way that cartoon wants to show you. There's direct killing with an NK cell, which is activated from the process, and monocytes begin to roll in there. But it's very endothelial toxic, that antibody. CDC is complement directed cytotoxicity, and that's the other problem that we hadn't anticipated. So not only just the raw antibody, but it's what it does to cause complement to go down, and complement kills cells, as you know, and targets MAC, which is the final resting place of complement, and it punches holes in your endothelial cells and kills them. This was the biopsy of the first one with all that complement in addition to the antibody down there, and then this was a picture of that nice endothelial cell here. That's a red cell in the middle of that capillary, but at this point it was still raw. It was still percolating, hadn't reached the point of killing yet, and probably because we have all that stuff on the endothelium that was engineered in excess, more complement, more thrombomodulin, that kind of stuff that probably mediated this, gave us more time, but wasn't enough. And then the second biopsy out when he was in trouble, shown here, lots of complement still, and now these endothelial cells are blowing up. I'm about finished, but there are a couple other really neat things I wanted to share with you. Cell-free DNA has revolutionized a lot of things. As you know, we use it for following patients in lieu of endomyocardial biopsies after transplantation. It's become standard of care in many ways. But you can also go deeper into that, and you can find out where that DNA has come from. You can find out what's the cell of origin, not just did it come from a donor, you know, so that you can actually keep refining backwards on that cell type that produced that piece of DNA. And so this is fragmentomic examples of cell-free DNA. This is porcine endothelial cell here in the left side. So, and it's day 30 is yellow when he got sickest, and the earlier days, day 7 is one of the darker lines. But you can see there's a progressive increase, right, in DNA being elaborated from endothelial cell source of a pig in our human over time. So it pretty much locks in that the problem appears to be right at the endothelium, and what that problem is, because we killed the virus out, right? That's not part of the game. It's got to be antibody-mediated rejection. So our tools and our reaction have to be to that if we're to go forward. We don't measure anything in this field in terms of effectiveness. All our immune suppression is shotgun. We don't know what the the levels ought to be of this, that, and the other thing. The first inkling of being able to measure whether we have blocked complement has come with this modified Ham test, and it's a lot complicated, and I think I've been talking too long, so I may not try to describe it, but just look at the lines of the graft. The red line is a cell that's been engineered to have very little complement defense. So if there's complement that's active, it's gonna kill that cell, and you can see that over time that red line goes quite high, and it reacts beneficially to the complement blocking. So with complement blockade, it gets up to luminescence, meaning live cells in this assay. And that means that there's plenty of blockade going on in the patient's serum. The other one there is that you need to look at is PIGA, which has been less well depleted of complement blocking in an engineered cell, and it's growing too. It's also going the wrong direction until we added eculizumab, which is a second agent in addition to the Bayonet as a complement blockade. So what we learned from this is we need dual complement blockade. Single blockade that we used in the non-human primates seems to be insufficient. The brown line that runs along the bottom is just serum C5b, which is just an example of the products of end stage complement activation. Almost last slide. This was a note that our last patient wrote to us. When he got sick, he was placed on ECMO to try to give us time to use plasmapheresis and really heavy dose anti-plasma cell therapy and try to turn his heart dysfunction around. After about 5 days, he was quite tired. This was the gentleman from the NIH. He knew what he was doing, knew what he wanted, and he said he probably had had enough. So we did an echo, and without us leaving the room, all the nurses were there and lots of family. In the echo proved that after this period of ECMO, the heart wasn't better. I told him, well, it's no better yet. He said, well, then I want to stop. And then he still had his tracheostomy, and so he couldn't speak well, and he's really breathless at that point. He wrote these words out. It took me a while to figure it out, and the sleuths among you will probably get it right away. But the first line's pretty clear. It says, I am sorry. It's the patient. And then ... I, and then the next line actually has been interpreted, I think accurately, I tried hard. And then the last line, he paused to deliver. And he did it with his index finger to read it like this. He says, I love you. So everybody was in tears. And I still am when I show this slide, because it's a very meaningful experience. We promised him a chance. We promised we would learn something. I think we've been good on both promises. We almost got him out of the hospital. We didn't anticipate what I believe to be treatable antibody-mediated rejection with very powerful drugs going forward. And we have a plan for the future. We're going to move in this direction with our next patient, given an opportunity to treat another patient. There is a company that will make very biospecific absorption columns. We're going to use their absorption column for IgG and IgM to get us through that initial phase where we might have preformed antibodies that are enough to start the process. We'll then begin to press down on plasma cells, which is the machine shop for antibodies, you know that. These drugs are coming out of multiple myeloma cancer therapy. You can use a couple of different types of drugs. I think carfilzomib is there. It's a protease inhibitor, and it's very toxic, so we probably won't go that direction. But the anti-CD38, the plasma cells of the CD38 cell that creates the antibodies. And it's immune to rituximab, which is our go-to B-cell killer, right? But rituximab does not hit that population. And then finally, we're going to go to really innovative new dual complement blockade. So we're going to hit it with therapeutic immune absorption, press the plasma cell, and we're going to do a better job on our complements. Our T regulation seems to be okay. We give a little tack, and we have accessory pathway blockade on board. But that should be all we need to do there. And hopefully we'll have a better outcome. And then my final slide may be that one. Bruce, do you remember that? You were at Hopkins at that time, and we were trying to figure out a way to preserve lungs for distant transport. You were observing at the time. So we've come full circle, and now you're observing something else altogether, and I'm deeply appreciative of you coming here. Thank you so very much.